Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi and a very good morning to everyone. Ladies and gentlemen, distinguished guests, colleagues and viewers joining us from all across the region, welcome to the IMR-SEAMEO Webinar Series 2026. I am Dr. Masita Arip, Head of Allergy and Immunology Research Center, and it is my absolute honor and pleasure to serve you as the moderator for today's session. Our focus today is incredibly vital and timely, titled Advancing Allergy Diagnosis and Research in Malaysia from Bench to Bedside. This webinar is proudly brought to you by the Allergy Unit under the Allergy and Immunology Research Center, AIRC, Institute for Medical Research, IMR. Here at the Allergy Unit, our ongoing mission is to bridge the gap between laboratory discovery and clinical application, ensuring that cutting-edge research directly translates into better patient care. Today, we are privileged to explore this journey from the laboratory bench right to the patient's bedside alongside two exceptional experts from our fields, Dr. Husna, Farhanah binti Ahmad and Madam Brenda, Leecyous. Ladies and gentlemen, this webinar series was initiated with the aim in mind to share the knowledge, discuss topics on biomedical research and present research outcomes. Before we begin the presentation, allow me to announce a few housekeeping messages from the Secretariat. We will not be taking questions during the presentation, but participants may post their questions using the link shared in the YouTube channel. The question and answer session will be conducted after both presentations have ended. An attendance link is also provided in the YouTube channel for those who would like to receive an e-certificate of attendance. Please subscribe to our National Institute of Health Malaysia YouTube channel and click for notifications whenever there is a new post. Ladies and gentlemen, for today's webinar, we have two speakers. Our first speaker is Dr. Husna Farhanah, the Head of Allergy Unit, ARC in IMR, with expertise in allergy and immunology diagnostics, infectious diseases and laboratory medicine. She obtained her Doctor of Medicine degree from Moscow Medical Academy and later completed her Master of Pathology in Medical Microbiology at Universiti Kebangsaan Malaysia. Throughout her career, Dr. Husna has been actively involved in clinical microbiology, allergy diagnostics, laboratory quality management, research and innovation. She also played an important role during the COVID-19 pandemic in Labuan, Sabah, particularly in molecular laboratory development, infection control and antimicrobial stewardship. Her research interests include allergy diagnostics, seafood allergy, antimicrobial resistance, tuberculosis and severe drug allergies. She has presented and published her work nationally and internationally, including award-winning innovations related to shellfish allergy diagnostics tailored for Asian population. For today, Dr. Husna Farhanah will be sharing her research experience with the presentation titled Building a Comprehensive Allergy Diagnostic Platform in Vitro, in vivo and beyond. Dr. Husna, the screen is yours.
Thank you, Dr. Masita Arip, for the warm introduction. I am Dr. Husna Farhanah binti Ahmad, Head of Allergy Unit from Allergy and Immunology Research Center, Institute for Medical Research. So, good morning to the esteemed audience from across all the SEAMEO region. I believe the audience is of mixed audience, whether it's from scientific background or non-scientific background. So, I will try to use as simple words as I can, and bear with me with some of the technical facts in the presentation. So today I am proud to share what we have been doing in Malaysia's National Allergy Referral Center. Since the year 2000, the Allergy and Immunology Research Center in IMR has served as the diagnostic backbones for our public healthcare system. Our journey began long before the molecular era, originating under the visionary guidance of pioneers like Datuk Dr. Shanah Murad and Dr. Masita Arip from serology unit to establish a dedicated allergy platform. So actually today we face a profoundly shifted landscape. We are witnessing a rise in allergic diseases that manifest as a rapid wave of allergic rhinitis among secondary school children and a high prevalence of food allergy among children. Concurrently, there are changes in landscape where we see cases of adult onset allergy, food allergy. So meaning our mother complain of skin redness and itchiness upon eating food that they used to tolerate previously. So one of the example of changing exposome are the climate change, where hotter and humid weather is the breeding ground for our house dust mite. And urban living lifestyle, such as Western diet and high fast food with high glycemic index, are directly driving the rise of conditions like atopic dermatitis among our young adult populations. Furthermore, nearly 90% of this young cohort had inadequate daily fruit and vegetable intake, where the socio-economic factor might be contribute to this issue. So this complex interaction in between our genetic, epigenetic, microecology, and environmental factor play important roles in the pathogenesis of allergic diseases. Allergy can be due to IgE mediated, which represent with rapid onset, or non-IgE mediated. As I will discuss mainly on IgE mediated, as it can lead to the life-threatening condition because of the rapid onset. So to understand the disease burden, our center conducted a 5-year retrospective audit. The results show a post-COVID rise in the atopic severity dominated by the respiratory problems, which include asthma and also allergic fungal diseases, while eczema and food allergy making up nearly a quarter of our total national diagnostic load. So facing this burden, the lab cannot just produce report. We must act as a gatekeepers for precise allergy testing. So talking about challenges in allergy care is actually really straightforward. The fundamental challenge daily in our laboratory is resolving the mismatch between sensitization, meaning you have the positive IgE antibody, with true clinical allergy. So meaning you can have positive IgE towards shrimp, but when eating shrimp, you have no symptoms. But in some of the condition, when you eat a lot of seafood, like in this picture, the symptoms appear. Furthermore, standard allergy tests are often complicated by non-specific interference and cross-reactivity. So to resolve this conflict, our platform must look beyond phenotypes to understand the precise pathogenesis or mechanism causing the disease. So to bridge this diagnostic gap, IMR, we apply an integrated diagnostic framework. So we never look at a lab result in isolation. As detailed in this workflow, we evaluate the clinical presentation first. Was the onset actually rapid or delayed? Is the pathway actually IgE mediated or non-IgE mediated? Are there specific cofactors present? From there, we continue with the testing. This tiered workflow is actually bound by the quality assurance to ensure every diagnostic output directly guides targeted treatment or safe lifestyle modification. So looking at the clinical presentation as a diagnostic challenge in allergy, like in the first picture, you can see that patient can present with just skin rash, itchiness and redness after drinking cow's milk. And the second picture, we can also see patient can give history of shortness of breath after exercising, but give history of prior intake of roti canai before exercise. And the third picture, patient can give history of skin itchiness and rash immediately after taking half-boiled eggs, but able to eat cake containing eggs without any symptoms. Thus, mapping out factors such as type of food, presence of cofactors such as exercise are necessary to guide us on further testing. So some sharing on the real case report. A 25-year-old female presented to the clinic with rapid onset of symptoms such as shortness of breath, skin itchiness minutes after taking the coconut pancake. So the clinical assumption might lead to permanently restrict this coconut and wheat and put the allergy label. However, when we evaluated thoroughly, the patient is not actually sensitized or have the positive antibody towards wheat or coconut, and patient actually previously tolerated well. So we actually examine the flour, and the auramide was microscopically seen in the flour that is used in the pancake preparation. So instead, patient actually had ingested high concentration of dust mite that had proliferated inside the flour in a warm, humid environment. So this case illustrates what clinical allergy actually is. It is where triggers, cofactors, and timing matter. So moving towards the diagnostic framework, I'll discuss about the test availabilities, quality assurance, and algorithm used in the allergy testing. As you can see, the allergy testing can be of in vivo testing, which is upon testing of the skin, in vitro, which use the extract base or component, and either singleplex or multiplex platform that we use. And to assess cellular, we can also use basophil activation test and biomarkers assessment. So currently, for screening, we evaluated allergy through in vivo skin testing and in vitro skin testing. So the current testing shown in this presentation, majority will focus on the IgE-mediated assessment. So looking at this slide, skin prick test is used to assess the presence of sensitization or IgE antibody through skin reactivity upon allergen application. So meaning this procedure is very simple. You just put the allergens on the skin, the culprit allergens, meaning some shrimp, if the patient is allergic to shrimp, put on the skin and prick it and wait for 15 minutes. If the IgE or presence of antibody, the skin will show an induration, like is shown in the picture here. From 1 until 6 is giving the positive reaction in the form of wheals. So while skin prick test actually remains a timeless and practical method, its clinical utility is actually limited to the extract quality and completely dependent on it. So due to this, our center and our institute identified some gaps in the availability of allergens from local food that causing allergy in Malaysia. Most of the commercial allergens for skin prick testing are manufactured in the North America or Europe and using their diets and based on their environment. But we do not consume or encounter the same food or the same environment as theirs. So to bridge this gap, our center pioneered the innovation project utilizing local food to make allergenic extract used in the skin prick test with the brand of My Allergen. So through the 7 steps of manufacturing workflow within our laboratory, these steps are actually carried out with the standardized method. So we actually, from 1996 to 2026, we actually consistently supplied this validated local extract to the major Ministry of Health hospitals across the nation, forming the foundation of more than 4,000 skin prick protocols. So apart from the in vivo utilization of the skin prick test, we do assess the delayed mediated drug or contact hypersensitivities via patch testing. So the suspicious drug will be patched on the back of the skin, and the localized dermal infiltration will be read after 48 hours. So moving towards in vitro, meaning you test the patient's blood in the lab. Okay. So many platforms available for IgE testing, whether it is automated singleplex, where we need to select each of the allergen tests based on the clinical presentation of the patients, or multiplex in the form of immunoblotting or the microarray, where panels of allergens were tested simultaneously against the patient serum. Decision on which platform to use should be based on the workflow, cost effectiveness, workload, and our demand. So performance of the test should be considered to ensure quality results produced, which I will discuss shortly. So looking at the cellular assessment, the BAT, which is the basophil activation test, is one of the tests that frequently considered as laborious. So this is a functional ex vivo test to assess the reaction of basophil to the allergen tested. and it can be used to test for allergens that are not available in the IgE panel or can be used among complex patients with multiple drug hypersensitivities. So finally, the emergence of biomarkers complete the profiling of the allergy patients. These biomarkers frequently are specific towards organ or system, as the underlying pathogenesis are much different. So this profiling is necessary for the precision approach in the treatment of the patients. So as part of building a comprehensive allergy platform, ensuring quality results that correlate well with patient is important. The quality assurance is divided into pre-analytical, analytical and post-analytical, meaning before we test the patient, meaning we see the patient, when we test the blood of the patient, and after we got the result from the analyzer. So before testing, these are the factors that we should take into consideration. Most of the clinics offer cheap price for more allergen tests or multiplex testing. But if we have no symptoms and results are positive, we will become very confused on how to approach these positive results. And sampling time do influence on the result. For example, when patient presented with bloody diarrhea after drinking cow's milk and blood is taken during the acute admission in the hospital, cow's milk IgE can become negative in the testing due to the IgE become consumed in the effector phases. So in this case, we will try to suggest for repeat testing after resolving the acute period. So looking at the analytical parts, as you can see here, decision of using any platform should be based on the knowledge of the principle and platform used. As you see, both of the diagram here, the left and the right side, are using different principles. And the assay design starting from solid phase or immobilizing phase should give less interference in the final results, especially in the quantitative testing. Hence, the evaluation should be done periodically inter-platform to ensure the results are not spurious, especially among complex or multiple positive patients. So apart from pre-testing and testing part, what we should do when we see the result is important at our laboratory phase. Frequently, the results are not as what the patient presented, especially among polysensitized or multiple positive antibody patients. So in this study, we conducted a study among 235 children with eczema, and 90% were actually polysensitized, predominantly towards food including cow's milk, egg white, wheat and peanut. This is difficult to correlate clinically, especially with kids cannot identify their triggers. So one of the causes of this multiple positive results, especially among highly prone allergic patients, and are caused by phenomenon of cross-reactive carbohydrate determinant, which is a short form is CCD. What are CCD? CCD actually is a sugar chain with epitopes attached to the proteins of plants, this is pollen, and invertebrates like house dust mite. So it has the ability to bind IgE independently of the allergenic proteins. Because this exact same sugar structure is similar, shared across thousands of unrelated species, the CCD IgE antibody can bind to almost any plant or invertebrate of the allergen extract that is used in the testing platform, giving false positive results. And furthermore, as you can see here, the testing platform that utilizes nitrocellulose membrane as the solid phase may contribute to the binding of the CCD IgE through their residual CCD from the cellulose matrix. Hence, this creates a diagnostic noise, a sea of positive results that often have nothing to do with a clinical allergy. So we conducted also a study among 300 patients with allergic diseases that show that we observe 1 in 5 patients actually are CCD positive with median IgE of 1,000 kilounit per liter. And this patient with high total IgE more than 1,000 were 5.3 times more to have positive CCD IgE. And this CCD positivity shift the diagnostic profile from specific to spurious. So 87.8% of these CCD positive are polysensitized, meaning they have multiple IgE results. And this patient are 12.4 times more likely to appear allergic to multiple unrelated allergen sources compared to the CCD negative cohort. Meaning, their IgE results are questionable. So in addition to that, cross-reactivity does play a significant role in our lab. In the study published by us on adult eczema, half of the patients actually are positive to our local extract, and about 98.3% had a co-sensitization with house dust mite and cockroach. This is a classic cross-reactive pattern driven by the tropomycin. We also uncovered important pattern in our study. On coconut, as coconut is usually deemed harmless. We evaluated coconut sensitization and found that 70% of the symptomatic patients were positive for coconut. And because coconut milk and peanuts are usually consumed together in our local favorite dishes like nasi lemak, 67% of these patients displayed co-sensitization with peanuts. So this co-sensitization make it difficult to distinguish a true primary food allergy from a harmless cross-reactive food allergen. So highlighting why advanced molecular profiling or component resolved diagnosis is necessary. So talking about utilizing the component resolved diagnosis, we can gain a clearer diagnostic information. Instead of testing against a broad crude extract, CRD allows to dissect the allergen source into its individual protein molecules. This diagram actually outlines the geographical variance in CRD within the Asia-Pacific and illustrate how local environmental factors, climate and dietary habits dictate distinct molecular sensitization pattern across different countries. And as shown in this picture, in Singapore and Malaysia, the molecular component is dominated by dust mite, the P, the F and blue T. In addition to this molecular resolution allows the precise clinical risk stratification. So look at the peanut component scale. If a patient has antibodies against RRH, the seed storage protein, which is the red color one, we are dealing with highly stable structures and resist heat and digestion. So this profile flags a severe risk of life-threatening systemic anaphylaxis. So to facilitate testing, IMR actually operate under the integrated algorithm, whether we use a top-down approach as patients comes and we take a detailed history with physical examination and screen with extract-based testing, and resolve the ambiguities with the component resolve before implementing the therapeutic plans. However, when patients present with highly complex history with multiple positive results, we usually use a bottom-up approach where we test molecular component to quickly identify and separate true triggers from the cross-reactivity. Finally, let's look at the future direction, next generation omics. Moving beyond the extract testing requires an integrated system of omics approach. So combining genomics, transcriptomics, proteomics, and metabolomics into a comprehensive network will define individual patient disease mechanism. So a key example of this current shift is bead-based epitope assay. Instead of evaluating the whole protein, we assess the linear peptide strand that will bind to the fluorescent bead, and this specific linear structures is mapped across major protein epitopes. Furthermore, using machine learning models, the area under the curve for epitopes vastly outperform traditional crude extract serology. We achieve lower area under the curve in diagnosing food allergy in these wheat allergy cases. This confirms that this linear epitope binding intensity correlates directly with the clinical severity in patients with true anaphylaxis. This computational approach represents the future of precision medicine in allergy care. So, my conclusion. If there is one central message to take away from this 24 years journey at the IMR, it is this: allergy diagnosis can never be reduced to a solitary positive result on this piece of paper. Okay, this is not just a piece of paper. And this diagnostic laboratory should not just be a testing center and produce report. It should be a platform where clinician and laboratory work together from the moment patient come to the moment patient go. So true precision can only be achieved when advanced molecular and cellular diagnostic are woven directly with patient's real history. So combining laboratory science with clinical context is the key precision to medicine. So with that, this is my contact information. And with that, thank you very much. I pass this floor to our esteemed moderator, Dr. Masita Arip. Thank you so much.
Thank you so much, Dr. Husna, for charting our unit's 24-year journey from 1999 to 2026. You have clearly shown how Malaysia's changing environment and modern lifestyles have driven up allergy cases, creating a massive rise in respiratory issues like asthma and childhood food allergies. It is incredibly proud to see how our unit successfully translated bench research to bedside care, notably through our in-house formulation and selfish extract supply. Okay. To Government Hospital Nationwide. Dr. Husna also gave us a vital roadmap on quality assurance, reminding us how to navigate tricky diagnostic interferences like cross-reactive carbohydrate determinants, CCD, and crustacean dust mite cross-reactivity. Ultimately, by introducing next-generation tools like component resolved diagnostic, CRD, and basophil activation test, BAT, you left us with the most important take-home message: sensitization alone does not equal clinical disease. True precision medicine cannot exist in a standalone laboratory silo. It requires us to weave advanced molecular diagnostics directly with the patient's real-world clinical history. If there are any questions that you would like to ask about her presentation, kindly submit your questions using the link provided in the YouTube channel. Next, I would like to introduce our second speaker for today is Ms. Brenda Leecyous, who is a research officer in Allergy Unit at ARC IMR, with over a decade of experience in biomedical research. Ms. Brenda is currently pursuing a PhD in molecular medicine at University Malaya. Her research utilizes proteomics and metabolomics to profile human milk during extended lactation, advancing insights into infant development and circadian biology. She is highly proficient in advanced analytical techniques like LC-MS, flow cytometry, and bioinformatics platform. Ms. Brenda has led and co-investigated multiple funded projects in food allergy diagnostics and translational immunology. Her award-winning work is widely published and presented at regional and international scientific conferences. It is our great privilege to have Ms. Brenda with us to share her research. Her presentation title is Allergic Diseases Research in Malaysia, Translating Diagnostics into Clinical and Public Health Impact. Over to you, Ms. Brenda. The screen is yours.
Thank you, Dr. Masita, for the kind introduction. Good morning, everyone. Thank you for being here today. My name is Brenda Leecyous. It is an honor to be here today to present at this IMR-SEAMEO Webinar session. Today, I will be sharing some allergy research work in Malaysia and also some updates in latest allergy diagnostic tests. So this is the outline of my presentation. I'll be talking about the journey of allergy research in Malaysia, briefly about the current allergy diagnostic landscape in Malaysia. Most of my presentation will be focusing on the research and some updates on the diagnostic tests, updated diagnostic tests, and I'll be giving some future approaches to allergy diagnosis and key takeaways. So before I take you through, I would like to start with some history of allergy research in Malaysia. The Allergy Unit was established at the Institute for Medical Research in the early 2000s. During this time, the allergy diagnostic were expanded. The allergy diagnostic tests were expanded, which include tests such as total IgE, specific IgE, tryptase, and also eosinophil. During this period, it had seen the establishments of the first allergy clinic in the Ministry of Health, Malaysia, within the Hospital Kuala Lumpur facilities, which laid the foundations for structured allergy services in Malaysia. So, and between 2010 and 2020, the research has focused to improve diagnostic tools for the local needs. These include developing diagnostic tools relevant to Malaysian, which involve work such as food allergen identifications and characterization. From 2020 onwards, the allergy research has become more translational, directing research into clinical applications. So some of the research work includes standardizing local allergen productions, explorations of component resolved diagnostic, which is a molecular-based allergy testing, and refining the drug allergy diagnostic. So today and into the future, the Allergy Lab continues to strengthen its role as a national referral center for allergy diagnostic. In allergy diagnosis, oral food challenge has remained the gold standard for food allergy diagnosis primarily. However, it is rarely performed in routine practice due to several challenges, which include the need for trained personnel, the requirements of specialized clinical settings, and these tests also time-consuming procedures which involve a risk of allergic reactions such as anaphylaxis. So the current allergy diagnostic workup include taking the patient clinical history supported by total and specific IgE, and where the testing done using both crude and molecular extract. This test has ECP will also perform to cater allergy-associated cases such as asthma and chronic rhinosinusitis. Other than the current diagnostic tools, together with the allergy laboratory worldwide, we also believe that there is more room for improvement towards developing more accurate, reliable, and accessible diagnostic approaches. So emerging diagnostic tools such as epitope-based assay, which has been described by Dr. Husna just now, and cellular-based assay may in the future reduce or eliminate the need for oral food challenge. So why a precise allergy diagnostic is needed in Malaysia? One of the many reasons is geographical variations in the distributions of allergens, especially aeroallergens such as dust mite, fungal, which is mainly affected by the tropical weather in Malaysia. These aeroallergens may lead to food cross-reactivity syndrome, which happen when IgE sensitization to aeroallergens first developed through the respiratory route and subsequently developing food allergy. This has complicates the food allergy diagnosis, leading to unnecessary food avoidance and mismanagement. So most of our current work focuses on the identification and characterizations of true allergens. Because testing using conventional crude allergens which contain many proteins, including the irrelevant one, that may lead to cross-reactivity and therefore cause false positive. So when we test with precise allergen using single allergen molecule, a well-defined allergen molecule is represented, which could reduce the cross-reactivity and therefore accurately define true allergy. So such testing allows us to identify the specific proteins causing the reactions and therefore gives a better patient management. So one of the previous study has highlighted the importance of considering geographic differences when diagnosing for allergy. In this study by Tam and Leong, Spanish patients were found to be more sensitized to lipid transfer protein, also known as RIH9, and developed peanut allergy later than American subjects. So in this group of patients, allergy reactions tend to occur after initial sensitization to plant-derived food. So in contrast, majority of American patients were sensitized to RRH1, 2, and 3, which are seed storage proteins. And this group had an earlier onset of peanut allergy with more severe clinical symptoms. So the sensitization rates to birch and grass pollens were also very low in this group. So meanwhile, in the Swedish children, they have the highest sensitization rate to RRH8 and its homologue, which is the birch pollen allergen BET-V1. This is an example where sensitization profile could vary between countries. The differences between these countries could be influenced by different environmental exposure and also unique dietary within this different population. So as part of our mission to improve diagnostic precision, we are currently working on developing and adapting more precise diagnostic tests, especially by optimizing and refining the allergens used in allergy testing. So our effort includes standardization of allergen extract. For this effort, the Shellfish Allergen Panel for skin prick test was introduced in 2024. We are also working on to establish a functional cell-based assay, where we have started with basophil activation tests for both food and drug allergy testing. Other than that, we also focus on understanding and comparing allergic disease mechanisms across age group, populations, and different regions. So in 2024, the Shellfish Allergen Panels called My Allergen were introduced through collaborations between Institute for Medical Research, University Pendidikan Sultan Idris, and Biomed Global. This panel consists of 5 types of shellfish extract that are commonly reported as the major cause of food allergy in Malaysia. The selection of these 5 allergens is based on a previously done research and experimental investigation. For your information, this panel is currently undergoing standardization process and they are yet to be commercialized. So these are the preparation steps of the local seafood allergens. We first start with the preparations of raw allergen materials. We source our raw materials mostly from local wet market or supermarket. After cleaning and weighing, we perform a physical homogenization, followed by several ultrafiltration steps up to 0.2 micron. And then we aliquot the filtrate into a small volume for lyophilization. For quality control, we perform SDS gel check, total protein estimation, and immunobloating to check for IgE binding properties. The lyophilized powder is kept in -20 for prolonged storage. So whenever there is a request from hospital or clinic, we will reconstitute the allergen up to a certain concentration before they are being transported to respective clinic and used for skin prick tests. So we mostly prepare seafood allergen extract, which include fish, clams, cockle, blue crab, and several types of prawns. We also prepare allergen from beans such as yellow dal, red dal, soybeans, and some fruits such as melons and tomatoes, and chicken and some kind of red meats. So moving on to talk about the emerging functional cell-based assay in allergy diagnosis. The currently available functional-based assay involve interrogations of allergy effector cells such as basophils, mast cells, and T cells. For all these tests, allergen stimulation will be performed, followed by measuring the expressions of biomarkers that require flow cytometry. So the BAT is an assay that measures expressions of activation markers such as CD63 and CD20C on its surface. BAT has previously been shown to be more accurate than SPT and specific IgE tests in assessing reactivity threshold. In BAT, the cross-linking between allergen, IgE, on the Fc epsilon receptor on the surface of basophils will induce the receptor aggregations and intracellular signaling. So this leads to the activations of calcium channel and release of lipid mediators and cytokines such as IL-4 and IL-13, which is responsible for the clinical manifestation. So the concentration of allergen-specific IgE, IgE affinity, and the ratio of allergen specificity to total IgE will determine the release of these mediators out from the effector cells. So Radulovic et al. In their study has demonstrated that BAT are able to differentiate between true allergy and sensitized patient. The study investigated whether the BAT could predict, one, the severity of allergic reactions. The second is the threshold dose that trigger the reactions. So in this study, the author recruited 150 children, subject them for oral food challenge, where 22 children reacted to back milk, 7 with severe reactions, 15 with non-severe reactions. And on the other hand, 49 children reacted to fresh milk, 12 severe, 37 non-severe. They found that BAT is the only biomarker that could differentiate severity and threshold groups. The severity cutoff for back milk is around 57% of activated basophils, and the threshold cutoff for fresh milk was around 1% of basophil activation. This suggests that basophil, BAT, may not only diagnose food allergy, but also help to predict reaction severity and threshold in children with milk allergy. In our setting, we are currently adapting a BAT protocol by Bowman Flowcast. We also incorporate the local allergen extract with BAT, with the vision to offer precise ex vivo testing for patients who are unable to undergo SPT or provocation tests. Other than that, we also use BAT to diagnose drug allergy patients, where we are currently assessing its usage for LA drug. So I'm sharing here is our first experience using local allergens to compare between BAT and SPT. In this study, we compare SPT and BAT in 15 patients and 7 controls. When you look at the SPT results, crabs show a higher positivity rate at around 53% compared to prawn at 38%, suggesting that more patients were sensitized to crab based on skin testing alone. So when we use BAT, the positivity rate increased substantially for both allergen, 76% for prawn, 84% for crab. So this suggests that BAT may be detecting sensitization that SPT is missing. So the key takeaway here is that BAT consistently identify more positive cases than SPT for both allergens. This raises an important question. Are these patients truly sensitized but SPT negative, or BAT is picking up subclinical sensitization? So this warrants a further investigation. Importantly, all controls here were SPT negative, and BAT stimulation's index value has remained low, which confirmed the specificity for both tests. So now we move on to mast cell activation test. The principle of mast cell activation test, also known as MAT, is pretty similar to BAT, which is that requires the activations of the cells. MAT uses plasma to activate the mast cells, which do not belong to the patient being tested. It is usually derived from blood, progenitors, or mast cell line. The MAT is often used to address the limitation of the BAT, such as the need for fresh blood, and in patients with non-responsive basophils. This is because around 10% of the patient do not respond to basophil stimulation, which is also seen in our lab. In these patients, MAT can be considered as an alternative approach. So When we compare MAT and BAT, the key difference between these two tests is MAT, the mast cell line, is passively sensitized with suspected patient sera. Meanwhile, in BAT, the whole blood of the suspected patients is directly stimulated with allergens. In both assay, the CD63 markers is expressed on the surface of the cells and is measured. For T-cell assay, T-cell-based assay is less common assay in the context of allergy diagnosis. This test is usually used to differentiate clinical phenotypes. In an experiment involving peanut allergy, there are two different T cells identified, T helper 2 cells and T helper 17 cells. These two groups of cells have different clinical responses in oral immunotherapy. One downside of T cell is that their responses do not reflect clinical reactions to allergen during oral food challenge, which could be due to the T cell responses occurring much more upstream during allergic reactions compared to basophil, mast cells, and also specific IgE. So beyond diagnostic, our research also include disease profiling during early life. Dr. Husna is currently leading a study on gut microbiome composition in children. This study aimed to determine the association between gut microbiome and immune development, where the stool from children was subjected to microbiome global profiling in a longitudinal manner. So this allows us to track dynamic changes in the gut microbiome as children grow and to understand how these changes are linked to the immune system development. We are also involved in investigating the seafood sensitization profile among AD patients in Sarawak. This study is part of the effort to address the importance of region-specific exposure patterns. In this study, the suspected AD patients were subjected to skin prick test, specific IgE, and BAT. We hypothesized that region-specific exposure patterns play a key role in sensitization leading to AD. The results from this study will give us some perspective on the region-specific exposure patterns and support the improvement of care for AD patients. Allergy diagnosis is moving towards more precise and patient-friendly methods. So in the future, we look forward to use alternative biological samples such as buccal swab, saliva, and stool, which are less invasive and more accessible for patients. And at the same time, novel technologies, including multi-omics, are helping us to understand the mechanism of allergic disease. In addition to that, integration of clinical and laboratory data using the AI and machine learning may further improve diagnostic accuracy and support better clinical decision making. For key takeaways, allergic cases are increasing and have a major impact on patients, affecting their quality of life. Accurate diagnosis is very important, as misdiagnosis can lead to unnecessary dietary restrictions, whereas misdiagnosis may result in severe reactions such as anaphylaxis. However, the diagnosis of allergic reactions remains challenging. The new tools such as basophil activation tests and peptide-specific IgE are improving the accuracy of and may reduce the need for oral food challenge. So looking ahead, combining new technologies and AI-based approaches will help to improve diagnostic precision and patient care. So these are the reference of my presentation. With that, I thank you for your attention, and these are my contact number. I will pass the screen back to Dr. Masita.
Thank you so much, Madam Brenda Leecyous, for that highly engaging presentation on how we translate advanced allergy diagnostics into real-world clinical and public health impacts across Malaysia. Madam Brenda beautifully illustrated the growth of IMR's allergy unit from expanding basic testing in 2001 to our current status as a national referral center and training hub. She explained the critical differences in the allergy landscape, showing how conventional crude testing can cause confusing cross-reactivity and false positives, whereas precision component-resolved diagnostics offer clear, accurate answers for personalized patient care. A massive highlight was her discussion on modern functional cell-based tests being advanced right here at IMR, specifically the basophil activation test and the mast cell activation test, which are revolutionary because they can actually differentiate between the severity and threshold of an allergic reaction without putting the patient at risk. Moving forward, Madam Brenda left us with a truly exciting look into the future of less invasive diagnostic using saliva or stool samples, alongside the integration of multi-omics and artificial intelligence to improve diagnostic accuracy. Her core message perfectly aligns with our session today. Food allergies are impactful, but by moving away from stand-alone laboratory silos and embracing innovative biomarkers limited directly to a patient's real-world history, we can achieve true precision medicine and better patient outcomes. We will now be moving to question and answer session. Please do not hesitate to send us your question using the link provided in the YouTube channel. And I will try to my best to connect you to our distinguished speakers through the posted question. Nevertheless, should the time does not allow us to address all the questions, our speakers will try their best to answer the questions through email. For Dr. Husna, we actually have one question from the audience. Yeah. Why certain patients only have allergy reaction during pregnancy? Over to you, Dr. Husna.
Hi, thank you for the questions. Okay, certain patient can have allergy reaction during pregnancy because actually our immune function or system during the pregnancy has a little bit has some physiological changes to adapt to our the baby that we are carrying in our uterus. So part of it, the allergic reaction is because of the physiological changes that happen during the pregnancy. So I hope that assists with my answer assists you.
Thank you, Husna, for the excellent answers. For Miss Brenda, also a question from the audience, from Rania. Hello, I want to ask, an asthma person can cause because they are allergic to dust mite, which their airways are getting narrow, that's why they cannot breathe properly. But when they laugh a lot or run too fast, they also get asthma. Is it because their cobalt cells produce mucus? Thank you. Do you want to answer this?
Thank you for the questions. Thank you, Dr. Masita. Asthma in general is the cause of asthma in general can be due to IgE mediated or non-IgE mediated. So answering from IgE mediated perspective or allergy, running or exercising does not cause allergy, but it could promote allergic disease through changing by dysregulating the immune balance and promote a microenvironment which favors IgE-mediated allergic responses. Besides that, these activities related inducing asthma can also perhaps impair the epithelial barrier functions, making it easier for allergens to penetrate. So overall, these activities could act as an amplifier. It can worsen or reveal the underlying sensitization rather than being a primary cause of allergy in most cases. Thank you. I hope I answer your question.
Thank you, Ms. Brenda. So now, Dr. Husna, second question for you from the audience, yeah, from Nur Hasana. My son has been on certain medication for 2 years, Rinvoq here. Previously, he was allergic to many foods like meat, white rice, and flour. We recently repeated his allergy test, and everything came back normal except dust mites. However, he still gets severe flares after eating meal. May I know why? Does Rinvoq mask allergy test results? Dr. Husna?
Okay, thank you, Dr. Masita. And Puan Nur Hasana, okay. Actually, in atopic dermatitis, you mean severe flares? I'm not so sure the question. I think Rinvoq is actually for the disease immunomodulating purposes for severe atopic dermatitis. So in atopic dermatitis, basically, you can have multiple underlying mechanism that are causing your flares. It's either it can be of the IgE mediated and also non-IgE mediated mechanism. So my answer is that you can consult with your allergies, and the testing of the test that show negative is actually of the allergy IgE mediated test. It's not the non-IgE part. Okay, that's all.
Thank you, Dr. Husna. Now we go to the second question for Ms. Brenda. For my allergen product from IMR, other than shellfish panels, do you have any plans to expand to other foods?
Thank you for the questions. If we have any plans to expand my allergen, the answer is yes, of course. That's the long-term plan for my allergen, is to expand beyond shellfish panels. Besides shellfish, we're also interested in extending the platform to other important local food allergens such as fish and milk. The ultimate goal is to build the region-specific allergy panel that support the accurate diagnosis that better reflects local diet. I hope I answer your question.
Thank you, Ms. Brenda. Now we have another question for Dr. Husna from the audience, from Nur Azia Hospital Kemaman, Terengganu.
Okay.
Quite far, not only in Klang Valley. We also have questions from Terengganu. A patient has multiple drug allergies and was afraid to re-challenge. Can doctors refer this patient to do drug allergy tests? If yes, where can the patient go to get drug allergy tests, government or private? And what is the cost for the test?
Okay. I'll answer it on behalf of our center. So our center is actually a diagnostic center, but we also do a collaboration with Hospital Kuala Lumpur, and we have a dermatologist team with allergies, which is Dr. Faizal in Hospital Kuala Lumpur. So you can refer to dermatologist Hospital Kuala Lumpur for drug allergy test, because they will do a comprehensive test and assessment there. Thank you.
Thank you, Dr. Husna. So now I think we have to ask the final question from the audience, Dr. Husna, from Dr. Ahmad Fairuz, MIOSH, from OSH, I assume. Do you do for chemical allergy tests? That's the question.
For the time being, we actually offer IgE testing, meaning allergy IgE testing for inhalant, food, and some of the medications, including the component. So we actually do not offer chemical allergy tests.
Thank you, Dr. Husna. I'm afraid that is all the time we have for the question and answer session. Any additional question will be answered by speakers through email. We hope that this webinar has shed light on some research work that we are conducting in IMR and would spark interest and could initiate research collaboration with others. Ladies and gentlemen, As we come to the end of today's session, I want to express my deepest gratitude to our exceptional speakers, Dr. Husna, Farhanah and Madam Brenda, Leecyous, for sharing their groundbreaking insights into advancing allergy diagnosis and research in Malaysia. From laboratory breakthrough to real-world patient care, it is clear that moving from bench to bedside requires continuous innovation and strong clinical collaboration. Thank you to our wonderful audience for your active participation. Don't forget to subscribe to our NIH YouTube channel. I would like to announce that we will have the next session of IMR-SEAMEO Webinar series on 26 June 2026. So mark your calendars. The theme for the next webinar is Rare Inherited Metabolic Disorders in Malaysian Children from Diagnostic to Molecular Insights. We hope you can join us at the next session. Please remember to complete the feedback form in the chat link to claim your e-certificate. Stay safe, stay healthy, and have a wonderful weekend ahead. Thank you.